Everyone, and welcome to our video on transpiration. So this is the fourth in our plant transport series, and what we're going to be looking at in this particular video is the spec reference 3.1.3D, transport of water into the plant, through the plant and to the air surrounding the leaves, and this is obviously going to be a focus, as the name suggests, on transpiration. First thing then, let's recap what on earth we mean by this word transpiration. And we're going to get picky now, okay? So when we are defining transpiration, we need to say that it is the loss of water vapour from the aerial parts of the plant. The aerial parts of the plant are the leaves, and it's got to say water vapour. If you just write down the loss of water from the aerial parts of the plant, not getting it. If you were to say it's the evaporation of water from the aerial parts of the plant, fine. If you were to say it's the evaporation of water vapour from the aerial parts of the plant, no mark, because we can't evaporate a vapour. So wording is key. I would suggest you just learn this definition, loss of water vapour from the aerial parts of the plant, and then you're guaranteed to get the marks. So when we actually consider how this water is lost, then if we think about our leaves, we've always got two sides, upper and lower side. Now we're going to reduce the amount of water we lose through that upper surface of our leaf because we've got that waxy cuticle. So that layer of wax that lies across the surface of the leaf on that upper side is going to restrict the amount of water vapour that's going to exit the leaf. Most of the water loss is going to occur through that lower surface of the leaf and the structures on that part called the stomata, those little pores. Hopefully we do remember from our GCSE and the work we've done in class that what we're looking at with those stomata are little pores controlled by the guard cells that surround them, and the number of stomata that open will be affected by light intensity. So what we actually find is that because high light intensity leads to more stomata being open, then we get more transpiration happening during the day when obviously it's light. If we now think about how we're getting this water to the leaf, as we've said previously, then it obviously enters at the roots and then travels up through the xylem. So remember, water in xylem. So it's going to go all the way up the xylem to the leaf. When it gets into the leaf itself, then the water is going to pass through the spongy mesophyll cells by that process of osmosis, so moving down that water potential gradient, and then it will evaporate from the cell walls of the spongy mesophyll. Once we've got that water vapour, it's then going to diffuse out of the leaf through those stomata, and that is going to happen down the water vapour potential gradient. When we actually think about why this whole thing is important to our plants, what we're looking at is the importance of the transpiration stream. Why do we need our plants to have this constant movement of water from the roots to the leaves? And there's really four key reasons to bear in mind here. First one is in that water that's being transported, we also have those dissolved mineral ions. So it's a way to transport the mineral ions to the other regions of our plant. Secondly, it's providing the water we need for photosynthesis. Thirdly, that water is going to help to cool the plant down as it evaporates. And finally, it helps maintain cell turgidity. So that's going to be very important in terms of allowing our plants to maintain their vertical nature without any form of a skeleton. So we now need to consider the factors that affect our rate of transpiration. So really what we're looking at here are a few factors you've looked at GCSE, and then a little bit of a new factor as well coming into play. So our first factor that affects transpiration is going to be the light intensity. And quite simply, the higher the light intensity, the higher the rate of transpiration, because in greater light intensities, more of the stomata are open and therefore more water vapour can exit the leaves. Factor number two is temperature. The higher the temperature, the faster the rate of transpiration, because that rate of evaporation of water itself is going to increase, and therefore we get increased diffusion because our particles have more kinetic energy, because higher temperatures means more kinetic energy for those particles as well. So it's a double whammy on the temperature. One, we've got faster evaporation. Secondly, we've got increased diffusion due to that increase of the kinetic energy. Third factor, air movement. The greater the air movement or the wind speed, then the faster the rate of transpiration. And this is just down to the fact that we're going to be moving that water vapour away from the surface of the leaf, which means that we're going to maintain that high water vapour potential gradient. Because rather than having a situation where we've got our little leaf and then outside the leaf these little water molecules would accumulate, if it's a windy day, then they're all going to be blown away, and therefore we're going to have a high concentration inside, low concentration outside, which means that we've got that high water vapour potential gradient between those two regions. Another one we looked at GCSE was the humidity, and what we actually find here is that the higher the relative humidity, the lower the rate of transpiration. Go careful not to get into that little habit of going, oh, higher this, higher the other. With humidity, that is a measure of how much water vapour is already present in the air. So if we have a high humidity, we will have a lower rate of transpiration, simply because we're going to have a lower water vapour potential gradient between the air spaces in our leaf and the air outside, therefore reduces the rate of transpiration. Last factor that we're going to look at is the water availability, and quite simply, if we have a lack of water in the soil, then we will not have enough water to transport through our plant to maintain that turgidity. So as a result of that, what we're going to find is the leaves are going to begin to wilt, the stomata will close because the guard cells are no longer remaining open for those stomata, and because of that, we're then going to find a reduced rate of transpiration. The last thing that comes up to do with this particular topic is how we can measure transpiration, so practical investigation into the rate of transpiration. And the first thing to know, the bit of equipment we will use is this thing called a potometer. So we can see it down here. This little thing is called a bubble potometer. Now, quite simply, what we actually have is a little beaker of water with the end of our capillary tube just sitting in it. Then that capillary tube goes into this larger one where we've stuck our bit of plant in the end, our cut shoot. And our capillary tube is going to have a little scale that we can then use to read measurements. So what we're actually going to do then is we need to make sure that we cut our little stem underwater. That's going to be important because it avoids us creating basically an airlock. Because if we don't cut it underwater, what we're going to find is air is going to enter the xylem, and that's going to then affect our transpiration. So we need to cut it underwater first of all to avoid that issue. We then put it into our little end here, making sure obviously we've got a good seal around it, and we're then going to make sure we've got our air bubble in our capillary tube. And usually what we're going to do is make a little reading of where it starts. Now, depending on what factor we're changing is going to determine what thing is sitting here. In this example, we've got a light, so we're just going to be changing our light intensity. So we'd start with obviously our meter rule, working out how far away our light was initially from our plant. We would obviously then turn our light on, leave it for a certain period of time, and then record how far our little bubble has moved along the scale. What we can then do is we can turn our light off, move it 10 centimeters away, for example, repeat the experiment, record how far the bubble moves in that same time frame. Obviously, we can do a distance divided by time to actually get our little sort of rate of movement there. And this then allows us to compare the light intensity and how it affects transpiration. If you wanted to investigate the air movement, we would replace our little desk lamp with a fan instead, and we could obviously change the speed of the fan to get those different movements. We could do things like removing a few of the leaves if we wanted to see how the amount of stomata affected transpiration. there's a whole range of ways that we can modify, obviously, those factors. The key thing is how we could avoid there being more than one factor changed at a time. And an old favourite here is if we've got our light sitting close to our actual plant, we are getting a light, but we could also see a change in temperature because these little desk lamps do heat up quite nicely. So what we may well do is get a rather large measuring cylinder. So these are one of the big ones you normally only see in the prep room. We'd fill it with water and place it between the actual lamp and our plant. And this basically acts as a heat sink. So that's going to absorb any of the heat that's going to be crossing towards our plant. It won't get to the plant, so that won't change in temperature in any way, shape or form. But obviously we will just be changing the light intensity. As always, I suggest that you subscribe to the channel so that you can see when we next upload another video, and head on over to the A-Level Biology website where you can find a range of other resources all designed to help you with your A-Level biology studies.